In that case, it is my great pleasure and honor to introduce today's speaker, Ms. Dusti Shay, graduate student. Has a bachelor's degree in anthropology. Dr. Milanic, which I think that she likes teaching. And then she wanted to get a taste for her research. Contacted me back in, I think, 2016, asked to do some research lab, what I explained. Hey, but if you want to work for free, send me student for, I can do that. So not much has changed, but anyway, so our journey began together. And lab, she ended up doing her master's degree. More about. And let's see. So through her department TA shifts, which she teaching experience, she Dale Brigham Teaching Award in 2019. Realized early on the importance of writing and receiving grants. And her when she got her master's degree, actually wrote and received a small grant which funded her master's. Allowed her to develop some really savvy statistical analysis affecting the brain. So went and probably learned from brain affect the brain and do some space. That she did her master's degree in 2019 and then started her work toward her PhD. So she's had four publications, 3 first author publications. And then work at the obesity conference in 2019 in Las Vegas, I think is probably where I convinced her to do her PhD. Anyway, so I don't want to leave anything out, so I'm nervous. I want to make sure I get out. Really, I mean, so I think, you know, everyone has dealt with some struggles and certainly has experienced her fair share of challenges. And an inspiration to me in terms of going, and I hope she and most important, that's I know it's too. Most impressively, she just received an F-31. That through all of the challenges that she has faced, that's I'm just really proud of her, and I know that Mom and David are so proud of you. But without further ado, please join me in welcoming them.
All right, thank you so much. I'm excited to be talking about my research today. The primary thing that I wanted to talk about today is my F, the pre-doctoral grant that I just received. But since I was just awarded it in August, right, I haven't started, I don't have data yet, right? So I wanted to talk about the project itself rather than just the project. How did I get here, right? Why am I studying the brain? Why addiction, you know, all of the things, kind of my path. So not only that, but I want to be able to explain the science behind what I do too. So I'm going to start out talking about a lot of background information, things in the brain that are really basic, that kind of need to be understood to get the point of the experiments that I drew up my proposal on. And then I'll go on from there. So I want to start by asking the question, what is dopamine? Okay, dopamine is a catecholaminergic neurotransmitter that primarily functions in the brain and the central nervous system. It is produced from the amino acid tyrosine and then becomes a precursor to norepinephrine and epinephrine. Okay. Outside of the central nervous system, it also has functions in vasodilation, insulin secretion, sodium excretion in the kidneys, things like that. And the body really works hard to keep dopamine in this narrow balance. Too much or too little can result in some pretty detrimental conditions. Parkinson's, I think, is the one that's probably the most well known. It's a dopamine deficiency. And I'll talk about that more here in a moment when I talk about how dopamine works. But also schizophrenia and potentially ADHD are also conditions that are associated with dopamine. And so it really is impactful if something with dopamine changes, especially in the brain. So looking closer at dopamine, we have 5 dopamine receptors throughout the central nervous system, and they are categorized as excitatory or go receptors and inhibitory or no-go. And there's this whole go, no-go theory. And it's really the ratio of the excitatory to the inhibitory receptors that determines if we go or no-go. Okay, the easiest way to explain this, I think, is with movement. Okay, when we take a step forward, we have muscles that are pulling on our joints, but we also have other muscles that simultaneously have to relax. And that movement doesn't start in the muscles, it starts in the brain. And it's dopamine that helps control that essentially excites just the right ratio of go neurons to no-go neurons and produces a smooth and fluid movement. That's why we get the jerky, sporadic movements with Parkinson's, no dopamine present. But as I was explaining that, like just practicing it in my head, I'm thinking, how am I going to explain? What is reward, right? I don't study movement. If you know me at all, which I mean, most of you do, I don't study movement. So what do I study? I study reward. And what does that have to do with dopamine and movement? Well, it turns out that they're controlled in the same brain region, okay? The striatum is the largest nucleus of the basal ganglia in the brain. And the dorsal portion, wrong button, sorry. The dorsal portion, which would be situated right about here, is what controls muscle movement, eye movement, coordination, and things like that. The ventral portion is also known as the nucleus accumbens, and that's the region that I'm interested in. And the reason that I use movement as the analogy for dopamine is because traditionally we think of the neurons in the nucleus accumbens functioning the same as they do in the dorsal striatum. So we still have the go response and a no-go response. The difference is this isn't movement, right? This is rewarding behaviors and motivation to repeat those behaviors. And there was some research. I found one paper in particular published earlier this year that actually says we might not be able to trust the no-go theory completely within the nucleus accumbens. But there's just one study so far that kind of throws questions on that theory. So another thing that I want to talk about before I move on is 80% of the neurons in the nucleus accumbens are medium spiny neurons. That simplifies things quite a bit. Medium spiny neurons are named for their morphology. They're medium sized. They have lots of branches. That's why they're called spiny. And the nucleus accumbens receives input from various regions, primarily with glutamate as its neurotransmitter. It's an excitatory neurotransmitter. And dopamine modulates response. Okay, but what's important to know is that all of the efferents that come from the nucleus accumbens to other regions of the brain, they release GABA. And GABA is the primary inhibitory neurotransmitter in the brain. So specifically, the schematic when I found it, because usually when I talk about dopamine and reward, the first question people ask, or they try to relate to it and they think, well, I did that one thing that I really enjoyed, but I decided not to do it again. Okay. There's multiple kinds of choice that humans make, right? That are controlled by various brain regions that all go into these decision making. So when I talk about this is not a black and white thing. This is one part of a whole system, right? So usually when we read about dopamine, or we think about dopamine, we think of it as the pleasure hormone, right? The happy hormone. Which if you do something that's a novel behavior, you eat a new food, or I hate to use sex and drugs as an example, but those are also rewarding behaviors. You do something new and it excites you and you want to do it again. Okay, dopamine is responsible for that novel excitement. But after a while, after repeating those behaviors, the dopamine response dampens and it's not as fun and it's not as exciting. But the difference is where dopamine really drives us is in what's called motivational salience. And essentially, it just means my motivation to do it again, right? So if I try that food and I really like it, the next time I see that food, I'm likely to reach for it again. The drive to reach for it is what dopamine is responsible for, specifically in the nucleus accumbens. All right, so where does estrogen come into play, right? The title of my talk was Estrogen and Dopamine. So I have two very, very basic schematics here of neurons in the nucleus accumbens, okay? They are split up primarily into the excitatory, or D1-like neurons, and the inhibitory, D2-like neurons. They both have on the cell body, they have glutamate and dopamine receptors, okay? Primarily D1 receptors. There's a very, very small percentage of neurons in the nucleus accumbens that express both. So it's really one or the other. And then down here we have the D2. And again, it's the same thing, just D1 and D2 receptors on the surface. And then there was a study earlier this year that really took time to map out where are the estrogen receptors on these neurons. And it turns out about 83% of them are along the axon and at the terminals. And so we have ER alpha and ER beta. We'll also have kind of the lesser known G-coupled protein estrogen receptor, but that one doesn't play as big of a role in the behavior here. So I didn't include it. But essentially, glutamate excites, dopamine, if it's present or absent, modulates the excitatory response. And then estrogen, in some way, which we don't know yet, also works to modulate that response to dopamine. Okay. And yeah, again, traditionally D1 drives us to repeat and enhances rewarding behaviors, and D2 drives us to decrease or avoid aversive behaviors. All right, before I move on, are there any questions? Please stop me, because I'm more than happy to stop and answer questions along the way. We don't have to save them to the end. Yes?
The for the estrogen in D1 and D2 are similar, or?
So it really depends on the basal state of the neuron. And honestly, specifically the D2 receptor affinity has been investigated. In the last couple of years, I found a few studies, and the affinity of the D2 receptor for dopamine changes whether estrogen is present or not. And it's not always in the same direction. And so that's still really a really muddy area. But yeah, how estrogen and dopamine help to modulate these responses really depends on the basal state of the cell in the first place, how excited or inhibited it is. Yeah.
ER alpha and ER beta, present.
So the nucleus accumbens itself is split up into core and shell. And the research is still trying to tease out exactly how they function differently. They do seem to have a different function, but the core seems to express a balance of ER alpha and ER beta, and the shell, if I remember correctly, has more ER beta, but I don't remember the exact percentage ratios.
Nice.
Yeah. All right. So how did I get here? When I joined Dr. Vera Potter's lab, she primarily studies adipose tissue and estrogen, right? Well, the estrogen thing had me hooked. And when I joined the lab as a volunteer and then as a master's student, and she said, hey, I'm kind of interested in the brain. Would you want to take on this project? I jumped on it. The brain has always excited me. So I was really happy to take part in that. And I want to take a step back, though. There was a student before I came in, Youngman Park, who some of you might remember, who did a study in rats that were bred for high capacity running or low capacity running, which if any of you know Frank Booth's lab, that's exactly what they do, right? And in the high capacity runners, there was this strong correlation, or a direct correlation, between the total running distance, the weekly running distance of these rats, and this excitatory inhibitory ratio that I was talking about earlier. And this was done looking at mRNA expression, PCR, but it was really this right here, this association between running and dopamine receptors that got us asking these questions about physical activity and how dopamine might play a part when I came into the lab. So my master's work was in an aromatase knockout mouse model. So the mice could not produce estrogen at all throughout their lifespan, not during development, all the way up until they died. And I really just did a characterization of what makes these knockout mice different from the wild types. And it turns out that the males here, I mean, energy intake, total and relative resting energy expenditure, and physical activity in the as measured like in the Met chamber home cage, they really don't differ between wild type and knockout. Wild type are the black bars and stripes are the knockout. But these females, the female wild types, which are all these gray bars, overall, almost in every measurement that we had, were different than all of the other groups. And then we look at the knockout female, and there's a significant decrease, especially here in physical activity. And I wanted to know why. Fortunately, I was able to get the internal grant that Dr. Vera Potter mentioned earlier from the DNA Core facility. And we were really lucky to be able to do some RNA-seq. So point out here again the decrease in physical activity. And if you're not familiar with RNA-seq, essentially we took the nucleus accumbens, we took the DNA and the RNA out of it, and we sent it to the core, and they told me all of the genes that were expressed in that region, thousands and thousands of genes. But what we did is we sorted through those and said, okay, which ones are different between the wild types and the knockouts? And we categorize them, right? The most different to the least different. And one of our top genes was PTS. And this here is a correlation matrix. The 0.88 is our R value, or Pearson R value, so a very strong correlation. And this down here is our P value, so it's really significant. And PTS, when I looked it up, turns out that it's upstream of dopamine synthesis. It's actually upstream, or it's involved in the production of BH4, which is necessary for all catecholamine synthesis. And so that told us that we were kind of moving in the right direction, asking this question about estrogen and dopamine. This led us, for our next study, to create a new mouse model. We worked with Dr. Cheryl Rosenfeld's lab and created a model that we called the DIRCO mouse model, or DRD1 estrogen receptor knockout. And essentially what we did, this is the D1 neuron, right, that I showed earlier, and we used the DRD1 promoter region to knock out just ER alpha. Okay, so the neurons still had ER beta, and this happened, like I said, primarily in the regions expressed DRD1 nucleus accumbens, but there's also going to be some other brain regions there. And I say all this because after we did the characterization of it, we were hoping to see differences in physical activity, and we didn't. And of course, that's how science goes, right? It's a little bit frustrating. And of course, the explanation that could be that estrogen is working with ER beta, right? Which I just showed you was still there on the neurons. Or it could be that estrogen is interacting with DRD2, 3, 4, or 5, rather than primarily DRD1, at least in that brain. So for me, what's next, right? How did I get into addiction science? After completing all of this work in the lab and really focusing on exercise, I was reading about addiction in my spare time. And it doesn't take long for the nucleus accumbens addiction connection to come up. And so my brain immediately connected those two dots. And I thought, is this the same thing? Is this the same mechanism? And that's what generated my idea for the F31. And so moving ahead, that's what I'm going to be talking about is the proposal, now the award of the research that I will be completing over the next year to fulfill the F31 grant. And I'm excited about it. So my first question was, how does stereotaxic microinjection of estradiol in the nucleus accumbens affect wheel running post-ovariectomy? I'm going to explain all of that. So we know from our lab and kind of just in exercise science in general that when we Ovax mice move their estrogen, they move less. They run a lot less. And that's pretty standard. I expect that to happen if I Ovax these mice, okay? We also know from the literature that if you systemically give back estrogen, they start to run more. But what I want to know is if I put estrogen just in the nucleus accumbens, is it still going to rescue some of the running deficits that we see after Ovax? And this is the first part of my study. Okay, this will be aim 1. Aim 2. Oh, I'm sorry. I wanted to explain stereotaxic surgery for a moment. So essentially, these mice we will expose the skull, right? We'll find these 2 regions. The nucleus accumbens is a bilateral brain region. And we'll take a tiny drill and drill a hole into the top of the skull and then insert these very, very thin cannulae straight down through the brain just to the level of the nucleus accumbens so that we can slowly drip or micro-inject estradiol just in that brain region. And we'll dose it properly so that it acts just in that brain region. This is an image of the optogenetic, if anybody is familiar with that, the light therapy in the brain. It's done in a fairly similar way. It's just I'm dripping a compound rather than exposing it to light. Okay, so for my second aim, I wanted to do the exact same thing, but I want to know how it affects preference for cocaine. And this is the part where I'll get a little bit more explanatory in these procedures because the test for cocaine preference uses a box just like this. It's 3 chambers that essentially look different, look and feel different to the mouse or the rat. And we put this kind of neutral chamber in the center so that they start out in a neutral position and then they can go freely into either side of the box. And when we condition the mice to do this, we will start, and this is after their Ovax, after their cannula placement, we will start giving them injections of cocaine or saline, and we'll alternate those. And every time we give them cocaine, they'll go in one side of the box. And every time we give them saline, they'll go on the other side of the box until they learn. When I'm over here, I get cocaine, and I get this really great dopamine reward. And when I'm over here, I don't. And at the end of those conditioning sessions, we put them in the box for a test, and we watch which direction they walk. This is all videotaped, and it's not just a visual observation, but essentially the more time that they spend on the cocaine paired side, the higher their preference for cocaine. And in the addiction literature, there is strong evidence that estrogen modulates that response to stimulant drugs. When estrogen is high, human women report greater euphoria, a greater energy boost. They feel better after using drugs when their estrogen is at its highest point in their menstrual cycle. And the opposite happens when estrogen is low. And we've seen that in the addiction literature, we've seen that over and over again in mice as well. And so my hypothesis here is that when we give estradiol into the nucleus accumbens right before we condition them for cocaine, they're going to have a stronger preference for cocaine in the end. We also will have 2 testing days in here, merely because we want to make sure that there's not any effect of estrogen itself. So we'll have 1 testing day where we give them estrogen, and then the next day we give them nothing. They don't get cocaine on those days. They just move to the side in anticipation of cocaine, which is that motivational salience that I was telling you about earlier. Dopamine still gets released, and they still expect the reward. Are there any questions? Yes.
Yeah, so it looks like on your day 1, they're both overactive and given microinjections, so the nucleus is not ever really seeing absence of estrogen. It's always going to be the rest of the body may not be.
So the cannula are inserted for microinjection, and that's just those little tubes. We just place them and they're cemented into the skull, and they're just there all the time. We're not giving them injection until day 8.
Oh, I see.
Yeah, so we're OVAX just so that we have one surgery day essentially. We'll OVAX and place the cannula, and then give it a week before we start. Taylor.
Real quick, I was just wondering, are these animals going to have like phytoestrogen free diet or some kind of thing?
You know, exactly. Yeah, that's always a concern of ours when we do studies because we study estrogen, right? Turns out, I mean, as long as you're not using a standard cow, it's well, from what the test diet people told me this summer, that as long as you're not feeding a standard chow, almost all diets are now phytoestrogen free. They just use the soybean oil instead of like the protein, and that's where the phytoestrogens are. So yeah, I'm happy about that. It's less of a concern for me later on. Yeah.
What is the success rate?
Really high, really high. This is a stereotaxic injection is a really, really common neuroscience technique. It's honestly, I hate to say it's almost outdated at this point because that we've gotten so much better at figuring out how to stimulate specific brain regions without having to insert a cannula that potentially damages tissue. Well, it's going to guarantee damage tissue, right? But causes some confounding complications sometimes. Anything else? So I recently went to Chicago and threw a machete for the first time. So I have the picture up there. I want to take a moment before I end. I think I put this slide in the wrong place about the study that we are currently doing. Yeah, I did. I threw it at the end instead of right before my thank you. So I apologize. The study that I'm doing right now, we have termed the BLISS study, short for beta ligand study. And it is not related to my F31 or my addiction study, but it is going to give us some exciting information about the brain. So just really quick, we have 4 groups here that we are going to be testing. They're all going to be female. We'll have a control group, a group that will receive injections, daily injections of CL, a compound that induces thermogenesis that we have in our lab before. And then this new ER beta ligand compound developed at Ohio State University that this is their name for it, OSU ERB12, ER beta 12. And then we'll have a group that will receive both. And after we induce obesity with high-fat diet, and we're going to OVAX some of the mice, not these mice, but these 2 extra OVAX groups that we have over here, we're going to get them nice and fat. And then we're going to treat them for 2 weeks with these compounds, one or a combination of these compounds. And we're going to assess body composition. We're going to assess their metabolism in the MET chambers, their movement, their sleep patterns, and GTTs, and essentially see if there's a synergistic effect, right? There's some really promising data from Ohio State that Dr. Chris Toss recently sent, and it looks good. It looks good as far as improving metabolism, improving insulin resistance, just with this ER beta ligand. And so we're hoping that when we combine these 2, it'll be even better. We'll be able to improve their metabolism and reduce some of their metabolic risk after the high-fat diet-induced obesity. My part of the study, I mean, we're doing the whole study right now, but the part that I'm really going to be honing in on after we've collected all the tissues are these OVAX groups right here. Essentially, we don't know if this ligand crosses the blood-brain barrier. And so that's going to be my job, is to take the brains and to figure out, did this ligand make it into the brain? And if it did, what did it do? And I'll specifically, most likely be looking at the targets that I'm interested in with dopamine, measuring tyrosine hydroxylase and dopamine receptors and things like that. But that's really exciting because this drug is novel and it's not been tested outside of Ohio State yet. And so we're going to be able to be the first ones to produce data from this, and that's pretty exciting. Are there any other questions?
Dusti, can you share just a little
Yes. So when we look at locomotor activity in the home cage, if you were here a few weeks ago, I can't remember his name. I just said it the other day. He gave the talk about sedentary behaviors. Say that again, James. Yes, Dr. Stoner. He gave the talk about sedentary behavior, and he talked about how there's movement that we do that doesn't really cause anything metabolically, right, or doesn't have enough benefit, and then we have exercise. Well, the same thing kind of happens in mice. When we look at their home cage activity, their Met chamber activity, that's just their daily movement and such. So it's different from wheel running, right? Their voluntary wheel running. And based on seminars that I've heard before and literature that I've read, I think that the motivation for these behaviors, or the drive for these behaviors, I hate to even use the word motivation, happens in two different brain regions. I more likely think that the hypothalamus is what is driving this kind of basal move around the cage, just general sedentary behaviors. Whereas wheel running and the reward, the dopaminergic reward from that is driven in the nucleus accumbens. Interesting though is that, again, if this crosses the blood-brain barrier, and we will have running data also from the Met chamber, so that'll be a correlation that we could make. But if OSU gets into the brain, turns out that it looks, in the literature, it looks like ER beta is what's responsible for driving preference for cocaine, ER beta in the nucleus accumbens. And so that excites me when I think back to my F31 study, is that I really think that I will see something, right, just by adding estradiol back to the nucleus accumbens, and that'll be exciting in and of itself. So does that answer your question?
Yeah, no, I
Okay.
think it's, and in like this show. Culture's work. It was looking at ER beta versus alpha, right? It was alpha that drove the locomotor activity. But
Yes,
we might see distinction
in the medial preoptic area, right? I think there was a study that, that I just read from Dr. Yes. Yeah, that is a great question, because I was not aware of the degradation of the blood-brain barrier from obesity.
All animals getting high fat diet.
Yes.
I'm just.
Yeah. I mean, what markers do you have in mind? Like, that's absolutely a great question. The good news, I mean, even if it's not something that we're capable or we're able to do, all of the mice are getting high fat diet, right? All of the mice will be obese. We expect the Ovax mice will be more obese, just because Ovax mice gain weight, regularly. But so I would suspect that if that is an issue, it's going to be equal across all groups.
Commenting on that, if you are trying to assess whether if they would go through what
Yeah, I would think so. That would be, that would have to be a future study, because I mean that's not one of our primary outcomes, right? Like the integrity of the blood-brain barrier isn't something that we were considering. So yeah, it makes absolute sense to have a non-obese control group. But yeah, that would have to, the study's already going, so we would have to be a future study. Yes, Dr. Parks.
How does estrogen that's in the blood get into the brain, or is it made in the brain?
Both.
Okay.
So circulating estrogen can cross the blood-brain barrier.
Is there a transporter, or is it?
I don't know. I don't know specifically. It's not been a variable that's been brought up in the literature, so it's not something I've specifically thought of. However, I mean, estrogen is produced in the brain. That was what my master's work was about.
Okay, the
Yeah.
enzymes to make it are there.
Mhm. But the stuff that's produced in the brain doesn't circulate in the blood. It just acts in a kind of a paracrine manner. So
And then
just
I'm
locally.
confused about, I'm missing something. So the CL compound, that's working peripherally,
It's
change metabolism?
it's a beta-3 adrenergic receptor agonist. Yes, so it is working in the adipose tissue
You're not
directly.
injecting it.
Oh, yes. I'm sorry. I'm so sorry.
Not that, can it get into the brain?
CL?
I don't know.
I don't know. I don't even know if anybody's looked at that.
And so you're thinking that that upper right-hand quadrant up there is gonna change metabolism peripherally? And
This
then
one? Yes.
your hypothesis in the lower left-hand quadrant, why is there a peripheral and a brain hypothesis in the same study? How are you thinking to put those together?
So it wouldn't be like in the same outcome, if that makes sense.
These are groups.
Right. We're comparing these groups for their adipose tissue metabolism. It's primarily just these groups. It's
ER beta, which is injected or is it?
it's actually oral gavage.
It's oral gavage.
Mhm.
And so you think it's gonna work peripherally on the adipose tissue?
Yes.
Okay, and then you're seeing if there's some complementary nature in the lower right hand. And so you're part of this. So this is a peripheral study.
Yes.
Your part is to look in the brain in one of these, the lower left hand guy, to see if it compound gets in the brain. Is that right?
Yes.
Okay.
To see if it changes behavior.
Yes. Yes. Because like I said, we'll assess wheel running in the MET chambers, and so we'll have some physical activity behavior as well.
The big picture study is about adipose tissue.
It is.
Okay.
Yes. Yes. Yes.
I was just wondering, if it's
I don't know. I don't think that Dr. Koss has given us any food intake data yet.
It's a good question. I don't.
It is a great
we
question.
do know it's CL, that CL does increase energy expenditure, and so there's a small increase in food intake. But
Mice are really great at balancing out their metabolism.
it's just the compensate for the increased activity probably.
yeah, and sometimes you don't see it. You certainly don't see a reduction in the loss.
I was gonna say, if you
Mhm. Yeah.
Always like that.
I know that's always the thing is whenever there's a question, there's multiple brain regions, right, that are attributable to these certain behaviors. And I would love to be able to collect all of them and assess them and find out. I know. I know.
Just CL, you said this twice now, maybe CL increases energy expenditure, but it doesn't increase activity. Right? That's a pure peripheral basal energy expenditure.
Thermogenesis.
Yeah.
Half-life of estrogen, like injection, how long it takes to get?
So the half-life is about an hour. It's not very long. And that's why I'm giving the injections so close to conditioning sessions. We're not doing any estrogen add back in this BLISS study that we're doing right now. It'll just be for my F31. But yeah, it needs to be given pretty close to when the behavior that you want to affect, that you're hoping for an effect is.
So just to understand the box experiment,
Mhm.
you inject 1 hour later, or and you within an hour you put him in a box and then you decide where you're going to go, basically.
Oh.
Or like in these situations here,
Mhm.
you're going to inject.
So we will give a brain injection of estradiol on this day. 30 minutes later, we will give like a IP injection of cocaine, and then we will place them in one side of the box and leave them in that one side of the box until they learn this is where I get cocaine. Yeah. And that's just during the conditioning. That's how you teach them to associate one side of the box with the other. And then on testing day, you put them in the middle and see which side they go towards.
Doesn't seem to take very long.
It doesn't.
About
There,
3 days.
Yeah, so essentially over the course of 3 injections, yes. Now, there can be variability based on a lot of different things, of course. And the problem that could arise from this, and this was something that was addressed in the grant as well, was how many brain injections we're giving, right? And here we're proposing to do 6 in a row. And at first they were concerned about that. Fortunately, though, I had some good mentors that study exactly this kind of thing in mice. Dr. Carmela Rochelle is the one that, and Matt Will, that's here at MU, have both done that number of injections before in a brain and haven't seen adverse effects from it. And so we were able to justify that. But more than that, I'd be really hard pressed to not do enough damage in the brain that might be why the behavior changes. But yes, so the problem is if we get to the end of these 6 days in the estrogen groups and they haven't been strongly conditioned yet, that's gonna pose an issue.
Every time I see a complicated study design like this over multiple days, I wonder how many you can do in a day in terms of the sample size. Are you going to be running waves of mice, or do you study one mouse for 15 days and then another mouse and another mouse?
No, it's waves, because I did behavioral assessments for my master's work also, and they just take all day. Because you have, 5 to 15 minutes per mouse just for the test itself, and that's not habituating to the room, and that's not cleaning up afterwards, so it is just a complicated procedure. But we could probably do, with conditioning is a little bit easier because you can have a mouse on each side, simultaneously. But yeah, I can probably do 4 or 5 tests in an afternoon.
Oh, and how many surgeries in a day?
Well, I'm going to be challenging myself with this Bliss study. So we're getting ready to do OVAX, and I will be doing 8 in a day. So I practiced the other day. I did that OVAX surgery in like 15 minutes the other day. So I think it'll be doable. Yeah. Yeah, it does. It does. Yes.
So is each mice always going to receive cocaine first or going to?
We'll alternate. Yeah. This was the timeline example. And so for some of the mice, we'll start out with saline and then alternate. Yes, Neil.
Maybe I missed it. There's a bilateral.
Mhm.
Bilateral injection.
Yes.
Okay.
Yeah, they'll have cannula placed on both sides. That's always a question that I have. It's not uncommon, especially in like electrophysiology studies, to just manipulate one side of the brain. And when it's a bilateral brain region, like I'm not convinced that that's enough. Well, if you were just looking at the neurons, right, that's fine. But as far as behavior goes, I would definitely want to stimulate both, because I wouldn't want that to be the reason that we didn't see behavior changes. Yeah. Yes.
Are you able to house the mice together? You have to have them separated themselves.
Singly housed is the best option, because with these little stylets that are on their head, right, they can mess with each other's, and usually they don't. Honestly, once I've been around a lot of these mice and rats that have these, and they usually don't bother them once they're healed, but just in case. And with residual effects of cocaine, aggression is part of the withdrawal, right, that come down process. And so I would just keep them singly housed to avoid them fighting. Well, these mice will all be OVAX, so they will all be low. Yeah, yeah. Essentially, so yeah, I am, like I said, because we've seen in the addiction literature that direct correlation with the level of estrogen and the effect of the drug, right? How much euphoria they get. Then I'm that with OVAX, we're going to dampen that response, right? And then when I give it back, that's when I hope to rescue it. Does that answer your question?
Yes, and then how much the dose relative to their normal levels? Those you gave in, like how much fold increases versus someone with a normal level or my normal level?
Yeah, so I am dosing like directly into the nucleus accumbens based on previous work. Obviously, it's going to be a very, very small dose. It's not going to be a dose that would be like circulating levels, right? Because we don't want all of the circulating estrogen dumped into the brain at one time. So it'll be titrated essentially, you know, very, very tiny amount. Yes.
Have you cross-studied that?
No.
This is pregnancy test.
Yeah, I mean, yeah, that's a great question. Honestly, I mean, historically, we all know that, you know, females of all species are left out of research because of this issue, right? Because estrogen in the cycle is so hard to track and to correlate with behaviors. So yeah, I haven't even considered branching into the pregnancy because that all the normal fluctuations are blown out of the water. So.
We're talking about studies that have looked at women over their menstrual cycle,
Mhm.
right? But I don't think looked at women addicted. What happens when if they become pregnant and when and I don't know that of any of
It's
them.
probably a very, very niche area of research if it exists.
How it's
Yeah. 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 Not any studies that I have read. Yes, absolutely. Actually, the data that's the most compelling to me is the human study that associates essentially if a woman comes into a treatment center for cocaine addiction, where she's at in her menstrual cycle associates with how likely she is to relapse when she starts treatment. And so if she's at a point in her cycle where estrogen is high, she's more likely to relapse after that compared to someone that comes in when her estrogen is low.
I have a couple questions in chat.
Oh, is it? Okay. Okay. Okay.
So I have a question.
Yeah.
Something into the nucleus. Expect to see changes in food intake or appetite with that, and how that would, play a question.
Yes. So the question is, since we're injecting estrogen directly to the nucleus accumbens, is that going to change feeding behavior? And definitely a possibility. I'm not going to have these on high-fat diet, and I say high-fat diet because it's more rewarding, right? There's more hedonic eating activity whenever there's high-fat diet because of the dopamine response, as opposed to their chow, where it's not really enjoyable, but they eat it to stay alive, right? And so I wouldn't expect, I'm not going to be assessing that directly, but I wouldn't expect to see much unless they had a high fat diet or something that was more satiating, because I think that those kinds of basal metabolic homeostatic behaviors are driven more in the hypothalamus than they are in the nucleus accumbens. That's my opinion.
In like Matt Will's question.
Yeah.
Really interested not in cocaine. Now is about opioid high fat diet.
Yes. So there is, Dr. Matt Will, who is a mentor on my F31 committee. That's the research that he does. He doesn't look at cocaine specifically. He uses heroin and morphine to look at opioid receptors in the nucleus accumbens also, and see how they affect feeding behaviors, usually for sucrose or high fat diet. And interestingly enough, one of his students is, well, the last time I spoke with her, so a few weeks ago, she's currently taking rats through extinction. So she got them addicted to cocaine, and now she's trying to get them off of it, right? To the point where they don't show a preference when you put them in the box. They don't spend more time on one side than the other, and that's called extinction. And her rats that are on high-fat diet do not want to go through extinction. Like, they continue to show a slight preference for the drug, which I believe for her it's morphine. And so we had some fun discussions about that and the possibility of the food, the high fat diet, kind of bumping up the baseline of dopamine, right? And the drive, I guess for them it would be the opioid response, but bumping up the rewarding response essentially, so they remain sensitive to the drug, if that makes sense. with, oh, I clicked on the, there we go. So big thank you to Dr. Vera Potter and the whole lab team that is helping me out with the Bliss study right now. Everybody, I mean, I presented some of my master's work, so everybody that helped me work throughout my master's. And yeah, I'm excited that I got to talk to you guys about my research today. So thank you so much.